Hello,
Okay.
viewers. Welcome, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagitya. Today's our eminent resource person, Dr. A.KAVITHA Madam Garu, Assistant Professor, Department of Botany, RBVRR Women's College. Madam has explained clearly about the Pteridophyta, especially Lycopodium, morphology, anatomy, reproduction, life cycle. So today, Madam is going to explain the another type study of the Pteridophyta. Please welcome, Madam. Share your screen and start your session. Please welcome.
Okay, sir. Right. Thank you, sir. Yeah, good afternoon to all the aspirants of CPGET 2024. So today we're going on for the next life cycle of the type study belonging to Pteridophyta, that is EQUISITUM. So just I'm start sharing my screen. Yeah, just I'm going to start sharing my slideshow. Yeah. Yeah, EQUISITUM. This is another type study what you have studied in your degree. And this is the classification, the most important point of EQUISITUM. This type study, it belongs to the division called as Spinophyta, class Spinopsida. Just like Lycopodium, how it belongs to the class Lycopsida. Here it belongs to the class Spinopsida, order Equisitales, family Equisitaceae, and EQUISITUM. So when we look into the plant body, as we discussed, all the pteridophytes, they have the dominant phase sporophyte. EQUISITUM, here the plant body is a sporophyte with a well-differentiated stem, leaves, and roots. So just here you can see the diagrammatic representation here, the image of EQUISITUM. Okay, so this is the EQUISITUM here, having the stem, branches, as well as the leaves. Now, so one more important point I want to tell you here is this EQUISITUM, it is commonly referred to as horsetail. Okay, so horsetail, so you can look into the diagrammatic representation. So it is called as the horsetail. Now, so we'll go on to the morphology here. The plant body EQUISITUM, it has an aerial part as well as the underground rhizome. So this is the aerial part of the plant body, which is lying in the air. Okay, that's why we call it as the aerial part. Whereas this is the underground stem, which is prostrate, and we call it as a rhizome. Okay, so the plant body, that is the stem part. Basically, we talk about the stem part. It is differentiated into aerial part as well as the underground rhizome. Now, when we talk about the rhizome structure, because it is a perennial, horizontal, that is, it is growing horizontally on the ground surface, branched, and it is creeping in nature. Whereas when we talk about the aerial part of the stem, it is herbaceous and it is annual. So majority of these species here, they are very small with a size of around 15 to 60 centimeters height and only 2 centimeters diameter. When we talk about the species of EQUISITUM, the most commonly species what we observe, that is EQUISITUM giganteum and EQUISITUM tilmacia. These are the two which are considered to be the highest species of EQUISITUM, growing to a height of 13 meters and tilmacia growing to a height of 2 meters. Okay, so these two examples are considered to be, that is EQUISITUM giganteum and EQUISITUM tilmacia. These two are considered to be as the highest growing EQUISITUM plant species. Otherwise, majority of them are very, very small. Now, next when you go for the habitat of it, the EQUISITUM, it is generally growing in the wet and damp habitat, especially among the banks of the rivers and the canals. But whereas some species of EQUISITUM, they are well known for growing in xerophytic conditions. The examples are Arvens, the Equisitum arvens, and Equisitum debile. These are the two which are the examples of the plant species, Equisitum plant species growing in xerophytic condition. Whereas another most important point here regarding Equisitum is this Equisitum, the species or the genus Equisitum is considered as the indicators of mineral deposits in the soil. That means to say, in the soil, wherever the mineral quantity, that is the minerals are available more in quantity, there these Equisitum can be profusely grown. We found them growing more profusely in that particular areas. So that's why these are considered to be, this Equisitum species are considered to be as a good indicators of indicators of the mineral deposits. Whereas when we talk about here, even as some species I'm showing their xerophytic condition, we can see the deposition of silica on the outer wall of the epidermal cells. Due to the deposition of this silica, the texture of the plant will appear very rough in nature. Okay, it is not smooth. It becomes very rough, and due to this deposition of silica here, and the deposition of this will help in giving protection to the plant body against the enemies as well as the pathogens. Next, we're moving on to in detail, the structure of the stem. So the stem, it is going to have, just now I told you, perennial underground rhizome and the upper erect annual shoot. The branching of this aerial part is referred to as monopodial branching. That is, it can produce only a single branch. And the shoot system is differentiated into nodes and internodes. So in the majority of these species, the shoots are alike and they are chlorophyllous. And at the tip of the shoot, they bear strobili. Now, sometimes the shoots are dimorphism. That is, they possess both fertile roots as well as the vegetative roots. Now, when we go for the anatomy part of the Equisitum stem, before that, I want to focus here of something. Now, so the others are fertile, unbranched, they are brownish in color, right? So the underground rhizome and the aerial axis, they appear to be articulated or jointed due to the presence of distinct nodes and the internodes. Externally, the internodes, they show the ridges, the longitudinal ridges and furrows, and internally they are hollow tube-like structures. Now, when we go for the anatomy part of this here, so here the external, the outline of this anatomy, we can see the presence of, it is not perfect circular. So we can see here the wavy nature or the ups and downs of this outline. Generally, they are referred to as the ridges and the furrows. So these are the ridges and these are the furrows. So due to these, what are these? Because why these ridges and furrows are? This is due to the stem is going to be differentiated into nodes and internodes. And at the point of internode, we can see the arrangement of the leaves. So due to this reason, the stem or the rhizome, it appears like the wavy in outline and ups and downs of this ridges and the furrows. And here you can see the outermost layer will be the epidermis, which is going to be surrounded by cuticle with a deposition of silica. And below this cuticle, you can see the parenchymatous tissue here. So this parenchymatous tissue is of two layers, the outer cortical region, that is the cortex is differentiated into outer cortex and the inner cortical region. So the outer cortex here, we can see the presence of the parenchyma cells, which are showing the deposition of the chloroplast, whereas that we call it as the chlorenchyma, whereas the inner cortex going to see the presence of the parenchyma cells with the wide intercellular spaces. And in the outer cortex region, just below the ridge, you can see the presence of a patch of sclerenchymatous tissue. So this refers to the presence of this sclerenchyma and the presence of the deposition of presence of silica and deposition of the silica on the cuticle and the epidermis, it clear-cut gives you that the plant, some species of this EQUISITUM is adapted for the zero fatigue condition. Now, next come to here, in this inner cortical region, there are the presence of some hollow spaces, which are called as vallecular canals. So this is a very, very important point here in the anatomy of the equisitum stem. These are the large air spaces or empty canals like or an open cavities in which the food material can be stored. So this is called as the vallecular canals. So we can see here, these vallecular canals are lying just opposite to the furrows. So this is a very, very important point. The empty spaces or the vallecular or the cavities which are lying just below or just opposite to the furrows, those are represented as the vallecular canals. Now, next lying to the cortical region, there comes the stela region with a single layer of lying endodermis and a pericycle lying in between that. On the inner side, we can see the presence of the vascular cylinder with the presence of the vascular bundles. And another important point here is the xylem, or we can call it as in the below the xylem vessels. So you can see here a ring of vascular bundles, that is the xylem and the phloem. Below the vascular bundles here, we can see the presence of, and again an opening or a cavity which is called as carinal canal below the protoxylem. Okay, that is below the each vascular bundle, that is below the xylem, that too especially below the protoxylem, we can see the presence of a small cavity or a canal which is called as carinal canal. So the question may be asked here, the cavities in the equisitum stem of stela region, they are called as carinal canal. And just you can see the position of this carinal canal. Carinal canal will lie in the same direction of the ridge. Okay, I told you that the outline of this stem is having ridges and furrows. Okay, the carinal canal lies in the direction of the ridge, whereas the vallecular canal, they lies in the direction of the furrow. So this point is a very, very important point to be noted for your exam. Next comes the central region, which is called as the pith. Okay, so that is the reason why here the stem, when we take the section cutting of the stem, in the central we can see a hollow space. That's what I told you here. Externally, the internodes, they have the longitudinal ridges and furrows, but internally it is hollow in nature, appearing just like a tube-like structure. So that is the reason why I told you the hollow region of the stem is due to the presence of the central pith region, and carinal canal and the vallecular canals gives the most space for the storage of the food material. Next one comes to the root. The root is a primary root. It is of ephemeral type, and it develops from the primary root. It develops the slender adventitious roots, which arise endogenously at the nodes of the stem. So when you take the TS of the root here, epidermis, cortex, as well as the stele. The epidermis consists of the elonginated cells with or without root hairs, and the cortex is extensive with outer cortex having thick walls, sclerenchymatous, and the inner cortex, which is made up of thinner parenchymatous cells. Whereas when you talk about the stele of the equisitum root, the stele is a xylem is of triarch condition, tri to tetrarch, and sometimes in smaller roots they are diarch condition also, and a larger metaxylem is present in the center, and the protoxylem facing towards the epidermis. So here in the root system, we can't see any pith region. So here this is a one. Okay, this is a large metaxylem. So here this is a tetrarch condition. This is a large metaxylem and the protoxylem facing towards the epidermis. And this is the phloem. This is a simple protostele condition here. This is a large cortex region. and the outer epidermis with the root hairs. Next, when we talk about the leaves of the equisitum, the equisitum leaves are very small, simple, and the scale-like. And here the leaves are generally presented as isophilous leaves. That is, leaves are equal in size and shape. And all these leaves, they are united at the part of the length and thus form a sheet around the stem. Now, the number of the leaves per node. At each node, all the leaves are getting attached. So, the number of the leaves will vary from species to the species. For example, here I'll take a Equisitum schaffneri. This is the species in which we can see the presence of a number of 40 leaves at each node. That is, the number of the leaves I told you, it varies from one species to the another species per node. Now, the number of the leaves at each node, it corresponds to the number of the ridges and on the internode, which is on the anatomy. Now, the leaves here, they do not perform any photosynthetic function, but the main function of these is to provide protection to the young buds of the node. Now, when we go for the reproduction point of view, now even just like Lycopodium equisitum also will reproduce vegetatively. And the vegetative reproduction here, it is carried out by the rhizome, which is the plant which is present underground. So at the time of, that is during favorable conditions, these rhizomes, they develop some tuber-like structures on them. These tubers are the one which are going to store the excess of food material. And these tubers, when they detach from the rhizome, they develop and forms, they germinate. These tubers, they germinate and develops into a new sporophytic plant. So that is how the equisitum is going to be reproduced vegetatively. Now, when I talk about the sexual reproduction, the reproduction here, the adult equisitum plant, as we told you, at the apices of the stem, it is going to develop the strobili. The strobili are the one which we call it as the sporophylls, the aggregation of the sporophylls. Now, on the sporophylls, that is the leaves which are there, the sporangia which are developed on the leaves, these are sporangia, they produce spores. So, there is a peculiar characteristic feature here in the equisitum that has to be very well noted. So, here, the sporangia here, they are developed on a stalk-like structure here, which we call it as sporangiophore. Okay, so this is completely an aggregation of the sporangia. Okay, so this is sporangia. It is going to have a stalk which is called as sporangiophore. Now, so we'll look into this. So the spores, they are produced within the sporangia. These sporangia, they bond on the stalk, which we call it as sporangiophores. Now, all these sporangia are aggregated and forming a compact structure, what we call it as the strobilus or a cone. Now, when you talk about the structure of the strobilus, they are terminal in position. That is the erect stem of the equisitum on the terminal position. We can see the development of the strobilus. So they are terminal and non-chlorophyllous axis. It is composed of the central axis with a whorl of sporangiophore. Now each sporangiophore, it is a stalk structure bearing an hexagonal peltate disc at its distal end. And under the surface of the sporangiophore, that is under the disc, we can see the presence of 5 to 10 elongated cylindrical sporangia, which they are hanging below the disc. So this is a peltate disc. This is a hexagonal peltate disc on the sporangiophore, and on this disc, we can see a number of around 5 to 10 sporangia. Now, these sporangia here, you can see they are elongated and they are cylindrical in shape. And they are hanging. We can see them. They'll be forming just like a hanging position attached to this disc. Now, Now, the development of this sporangia here, when we talk about the mode of the development is eusporangiate type. That it is not formed from a single initial. So that condition, what we call it as leptosporangiate development. The sporangia, which will be developing from a single cell or a single initial, that we call it as leptosporangiate development. Whereas here in the equisitum, the sporangia are developed from a group of initials. So that we call it as eusporangiate type. Now the superficial cells, which are adjacent to the original initial, they take part in the development of the sporangia. Now the sporangia, they're initiated in a single superficial cell around a ring of the young sporangiophore. Now the periclinal division of the sporangium initial, it forms an inner and the outer cell. Inner cell will develop into the sporogenous tissue. Now sporogenous tissue or the sporogenous cell, it functions as a spore mother cell, and this spore mother cell, it undergoes meiotic division and results in the formation of a spore tetrad. And all these spores here, which are developed in this sporangia, they are of same size and shape, and hence even equisitum is also homosporous. Just like, unlike Lycopodium, even Lycopodium, it is, Lycopodium is a heterosporous type. So there we can't see that even Lycopodium is also homosporous. Okay, it is going to develop the spores. The spores are released and these spores are developing into the gametophyte. Okay, the spores are germinating and developing into a gametophyte. So on the gametophyte, sexual organs are developed. So it is of homosporous type. So here also in the equisitum, the spores which are developed in the sporangia, they are of homosporous type. Now, the spores upon germination, it is developing into the next generation of the plant body called as gametophyte. Now, this gametophytic plant body, the gametophytic plant body here, it is going to bear two types of sex organs, which are called as antheridium and the archegonium. Antheridium is called as the male sexual organ, and the archegonium forms a female sexual organ. Now here the gametophyte, it is basically a bisexual, or we call it as a monoecious condition. So it is an homothallic. That means it bears both male and the female sexual organs. Now, although there are certain members which are dioecious also, they are well also reported, but majority of the equisitum species, they are bisexual, that is, they are gametophyte, they are homothallic only. Now, next comes to the fertilization, that is antheridia, which are developed on the gametophyte. It produces antherozoids, the male sexual gametes, and the archegonia, it is going to develop the female sexual organs. That is, the archegonia, it develops into the female gamete called as the egg. Now, the antherozoids, with the help of the water, they will reach the female sexual organ archegonia, and by the disintegration of the neck canal cells, the antherozoids, they move into the venter and fuses with the egg and develops into the zygote. So here, for fertilization, water is essential for fertilization. So the gametophyte, it will be covered with a thin layer of water, so in which the motile antherozoids, they are swimming and reaching the archegonia. So just now I told you, the neck canal cells and the venter canal cell of the archegonia, they will disintegrate to form a passage for the entry of antherozoids. Now these antherozoids, they pass through the canal and they'll reach the venter, where there is the presence of the female gamete called the egg. Now the fertilization will occur and a diploid zygote is going to be formed. Now the zygote again develops into the embryo. So generally there are more than one archegonia, they get fertilized on the prothallus. Now, so next I told a zygote. The zygote is going to develop into an embryo. Now, so what is this embryo? Embryo, it is a mother cell. It is a starting, zygote is the starting cell of the sporophyte, we'll say. So the embryo is considered to be as the mother cell of the next sporophytic generation. So unlike most pteridophytes, there are several sporophytes, they develop on the same prothallus. So as fertilization happened on the prothallus, even embryo is developed on the prothallus. So as there are many archegonia getting fertilized here, we can see the development of many sporophytes on the same prothallus. So how this embryo is developed? The zygote, it will undergo first transverse division and it results in the formation of an upper epibasal cell and lower hypobasal cell. The embryo is therefore, it is considered to be as exoscopic condition where the apical cell is directed outward towards the neck of the archegonium in polarity. And here the embryo is not with any suspensor present, that the epibasal cell and the hypobasal cell, they divide at right angles to each other to form the oogonial wall and a four-celled quadrant stage, and all the four cells of the quadrant, they differ in size and shape, and thus this four-celled embryo, it will undergo very much subsequent divisions will occur and first results in the shoot apex and the leaf initials. And this one of the cell of the epibasal quadrant and a portion of the adjacent quadrant of the hypobasal region, it contributes to the development of the root. So the first root develops from one of the epibasal quadrants and a portion of the hypobasal quadrant. As a result, the shoots grow rapidly. Later, the roots are growing directly downward and it is penetrating the gametophytic tissue to reach the soil, and a number of such spores may develop directly from a mature gametophyte. So here you can see the formation of the young sporophyte. So here this is archegonia, and here the fertilization has occurred. The egg cell getting transformed into the zygote, and the zygote here is developing into the embryo. So first the embryo is formed of a four-celled quadrant. Then this four-celled quadrant undergoes subsequent divisions and result in an embryo. Now here there is a several embryonal divisions are going to happen, resulting in the formation of here the root initial and the shoot initials of this embryonal axis will occur, and as a result, this is a sporophyte developed on this gametophytic tissue. So as there are many archegonia fertilized here on the sporophyte, on the gametophyte, we can see a number of young sporophyte developing. So thank you. So this is about so I'll just stop sharing. So this is about the life cycle of the EQUISITUM plant. So any questions we heard in this, please?
Yeah. So my dear friends, any doubt, Madam will going to explain once again. Is there any doubt?
So if you have any doubts in EQUISITUM, so the only questions what can expected here in EQUISITUM is regarding the vallecular canal and the carinal canal.
Yes.
And regarding the horse tails, the common name of the EQUISITUM. So that is the questions. Bottle brush also we call it as, or not horse tails, we call it as a bottle brush. Or sprouting brush we call it as. Bottle brush antaru. So e 2-3 questions they mostly ask in the competitive exam. All the others are not.
Vallecular canals and the second one is
carinal canal in the
carinal canals. So both the canals are present in the stem of the EQUISITUM. This is the most important feature of this EQUISITUM.
Vallecular canals lies just opposite to the furrows, whereas carinal canal lies just opposite to the ridges. So that point needs to be noted and that are important. That's it.
Yes.
And the anatomy and the reproduction, it is same just like the other type studies. So mainly the important points are these to be noted. That's it. Having the
Okay.
xerophytic characters, these 2
Yeah.
points and the common name. That's it.
Yeah. Okay, madam. Thank you very much.
Right, sir. Thank you, sir.
Your beautiful slides are shown, and also the presentation is very, very excellent. Each and every corner of this topic has clearly explained. So
Right,
thank
sir.
you very much. Our
Thank you, sir.
participants are very happy with your class, madam. Thank you very much. Tomorrow we will
Yes.
meet same
Yes,
time.
sir. Thank you very much, sir. Thank you, sir.
Right, madam. Right.